Okay, so now I'm going to introduce another term which is really important, and that is the term of RFLP. That stands for restriction, fragment, length, polymer fission, and um, and that is actually the difference between the restriction maps of two people. So that's really important. Um, the RFLP is a snap that is uniquely located in a restriction site. Okay, once again, it is uniquely located in a restriction site. So that makes the RFLP extremely unique, and it becomes a tool in mapping. It's one of the most unique features of a person's genome. So now we have snaps and we have RFLPs that become tools of mapping, right? So you refer to a movie, enjoy. So actually, if you think about it, the RFLP is a very unique biogenetic marker, and that's very important. So you see in figure 3.3 on page 55, uh, the link between the restriction marker and a phenotype marker that can teach us about the frequency of recombination. Um, so SNAPs and RFLPs are unique features of a good genetic map. Okay, so we're going to look for them, we're going to use them. They are important tools also in understanding the effects and function of mutations. So, for instance, if we want to compare a healthy person with a diseased person regarding their RFLPs and SNAPs, that would be key, for instance, to understanding the presence or absence of problematic loci, sites, right, that cause certain disease and traits. So that's already the utilization of the idea of SNAPs and RFLPs. If all the people that have a certain disease have certain SNAPs or RFLPs in a certain location, we can connect that to the idea that there's a disease or not. So let's talk about a bit about the implications. So basically there's two major ones, right? We just talked about one, identification of disease through such loci and the link to the phenotype. The second thing is the isolation of a gene. If the two loci rarely recombine, the restriction marker must be close to the gene or to the genetic map. So that helps us locate and isolate genes. So basically, the bottom line of this of these last few points are that if we have a disease-creating gene, they're going to be located between SNPs. All right, that's kind of the idea. Right. So you refer to a movie. I want to be clear that though I gave the example of a disease because it's easy to understand, SNAPs and RFLPs are very useful tools in mapping other traits as well. It's simply easier to understand with diseases. Of course, we can use RFLPs to link different species. Okay, So exactly like we can use it within the same species, we can use it to show linkage between species. But the RFLPs are a more unique and rare feature than snaps, meaning that using only RFLPs to conduct mapping would be limiting relatively to snaps. Okay, a person's unique combination of sites in a region, is called a haplotype. So a person has unique features in a certain area, and that's what we call a haplotype. So a haplotype becomes a term in a certain unit that we can kind of scan for unique features. Right, you refer to a movie. So in a way, we can see the haplotype as a mini genome. So the haplotype is best used to describe a region that includes genes or alleles of common feature and function. So we're going to look for those, segments, those whole regions that serve a certain function or a certain trait together. And if you think about it, and if you understood the idea of the RFLP, then that is a way to decide or to determine the link, the family link, the genetic link between persons. So for instance, if I want to prove that someone is or is not my son or daughter, RFLPs would be the way. So the use of RFLPs and SNAPs is what we call DNA fingerprinting. And if you've ever seen CSI Miami or any one of the CSIs, whatever, you've probably seen that they use DNA to link people to crimes or other possibilities. So what they are theoretically doing in the TV show is mapping one's RFLPs and proving that it matches the person and the samples from the crime site. Okay, so this is huge. Okay, you refer to a movie, enjoy it. So when we take a haplotype, the more DNA it has in it, the higher its C value is going to be. So C value of a haplotype genome is the total amount of DNA in it. Okay, meaning that it kind of indicates how much is it interrupted or not. Between organisms, we find that C value varies, and that shouldn't surprise you because we talked about the differences between species and different organisms in their genome before, right? The genome size is not necessarily a criterion for defining a eukaryote or prokaryote. So that's important. It is agreed, though, that the genome size has an effect on the complexity of the organism, meaning that the bigger the organism, the bigger its genome, and the more complex it's going to be. When you go past insects, the relationship between genome size and complexity is lost, meaning that at some point the differences are not big enough to create, let's say, a linear relationship or any noticeable relationship. It doesn't mean that there's not differences between those organisms. It means that it's not big enough to create a certain ratio that is apparent and significant. Okay, so you are referred to a movie. Once again, I hope you enjoy it. So it seems that the overall size of a genome has little to do with the number of genes. And I'm assuming that you can actually explain that, because we've talked about the amount of different functions, sequences that we have in DNA, meaning that DNA does not only code for protein. It has its control factors, it has its regulation factors, and so on and so forth. It has junk DNA, it has introns and exons, it has non-translated areas, it has coding DNA sequences, and it has non-coding DNA sequences, right, for proteins. So that's not new to you. So yeah, the size of the genome does not necessarily give testimony to the number of genes, and that's kind of should be obvious at this point. So the lack of the genome size and the genetic complexity generates a paradox that's called the C-value paradox. Okay, so you cannot judge an organism, so to speak, through the length of through the size of its genome. It also means that two different organisms may have the same genome size, but will have completely different, will be completely different in their complexity. The other option, logically, is that two organisms with different genome sizes may have the same complexity. Alright, so you are encouraged to go over the key concepts of page 57 in the book. And now we're talking about dividing DNA sequences into two major types. And this is a logical part. It's just logical because if you understand the idea of the nucleotide sequences, it makes sense to look for patterns, right? So we have non-repetitive DNA, which are unique sequences, and you usually have one copy in a haploid genome. And the second will be the opposite, a repeating sequence of DNA that is present in more than one copy in each genome. The second type, the repeating sequences, the repeating DNA, can also be divided into subtypes. So when you say something's repeating itself, to what extent? Is it moderately repeating itself, or is it highly repeating itself? Usually, when the repetition level extent is high, the sequences tend to become smaller. Okay. The ratio between the two types tends to vary. Alright, so the first type, the moderately repetitive DNA, include what we call transposons. Those are short sequences of DNA that can actually move from one location to another, or even copy their cells. The extent of transposons varies between organisms, And they're also called selfish DNA because it seems to be as if they serve only themselves and care only about themselves. Um, it seems at this point that transposons have no effect of contributing or affecting the organism's development. They may affect the gene's rearrangement and selective advantages, but it's not going to affect development. Alright, I'm going to stop here. We'll continue in our next video.